So, in this condition, we can redefine a little bit what is genomics, transcriptomics, and metabolomics, because they are the three main omics science that are currently used to discover these new enzymes. So, genomics is always study of genome, but what it is interesting is that they provide some new idea or new clues, okay, regarding functional association between genes based on what we call gene clustering. Because genomics, okay, gave genome sequence, perfect, and we can find gene sequence, okay, that encode for protein. But what it is also interesting now in genomics is how genes are organized on the genome, on which chromosome, and if you look at one chromosome in particular, does all the genes corresponding to one metabolic pathway are situated or localized on one specific chromosome, or are they distributed across all the chromosomes? So, in fact, both situations exist. But it was interesting when genes are specifically clustering. Means that they are on the same, okay, on this portion, okay, of the chromosome of one genome. This could be also really interesting. So, means that now genomics is not only sequence, it can mean also structural information of how the genes are organized on the genomes. And transcriptomics has been also successfully used to identify new members. And what it is interesting in this case is to develop specifically algorithm to study co-expression of several genes together. In order, because the idea is when you have a metabolic pathway, to be sure to have the end product, the cells need to coordinately express all the genes that are necessary to take in charge the precursors and to produce the end product. So by this way, if you have co-expression of genes, you may have more probability that the gene co-expressed could be part of the same metabolic pathway. Okay, so means that now transcriptomics are not only sequence of mRNA, okay, but also the idea is to compare how these sequences are co-expressed or not, depending of the situation, depending of the cell, depending of the situation in which you have put your plants before taking the raw material to make the transcriptome. And for metabolomics, it's exactly the same. So now you have different way to analyze metabolomes, means that it can be a list, okay, of metabolites, but it can be also classified by family, subfamily, and by this way you can have some idea of how the family, the metabolic family, okay, can be co-regulated, co-expressed too, and this is a way to see also the link between all the metabolic pathways, secondary metabolic pathways. So means that now with this type of new angle, vision of this omics science, we can have more information with exactly the same base of data. So it is always new algorithms that permit to explore always the same type of data. So this is why it is quite interesting to see and to develop. And it is what we call integrative omics-based pathway discovery. I will show an example, okay? Just to complete what it has been presented by the two conferences of last years, is that what is the situation now, okay? How many plants have been already sequences. We have genomic sequences and so on. You know that the start point was the sequencing of Arabidopsis thaliana. That was the first model plant that have the particularity to have a very small, the smaller genomes as we can have, only 125 megabytes for the size of the genome, and until 2000, sorry, until 2000, which was the first genome sequence published. It was in Nature at this moment because it was really exceptional. Okay, in 24 years now we have more than 1,482 other plants that have been completely sequenced. It seems not to be so much, okay, but in fact, what we observe, it is a very important increase, okay, in the number each year, okay, of plants that are novelty sequenced. What it is true is that mainly sequencing have been mainly done on animals and on fungus for different other reasons you can imagine, and plants arrive in the first position of the number of sequenced species. And if we look at what are in plant kingdom, what are the families or the classes of plants that have been more sequenced, the majority come from angiosperma. So means the plants that are interest for us for several applications, foods, obviously, but also pharmaceutical or specialized metabolite. And this is very interesting. But what we observe now is that also several other plants, particularly Bryophyta, for example, or Lycopodiophyta, that are what we call primitive plants, are also now increasing of sequencing because of the idea to know how this metabolic pathway have evolved during the time, okay? And because this is also a way to find if one type of enzyme may have some different type of activity. I show you that it's not so one by one, okay? One enzyme have only one activity. You may have one enzyme with several activities, sometimes more than 10 different activities. And this can be supposed, for example, okay, if we have a continuous view from primitive plants to more modern plants, okay, particularly in several enzyme family. So here you have this type of information that it is right interesting. For one plant, we may have one genomes, but we may have different sequence of the genomes. Means that they can be produced by several different teams over the world. And in fact, it is also interesting because the genome is the same, okay, but the annotation of the genomes can be different from one group to the others. This can also permit to rebuild new sequence genome by two type of annotation in order to be more close to the reality, okay? But because to have a genome, it's quite simple. You have a sequence, but to see in the sequence which part are gene, which are not gene, okay, and how organize the genes is quite complex. So sometimes depends of the methodology you have used to make some annotation of this genome that can differ from one team to the other. And this is why when you have cumulative number of sequenced genome, you have a higher number, quite around 4,000 instead of the number of the total plants that have been sequenced, for example. And this is interesting because sometimes it's not necessary for you to sequence again, okay, the genome of one plant if this genome have been already sequenced, but you can kept all the information and you can mix all this information to rebuild, okay, a more clean, precise, okay, genome annotation. This is also what we can do. And you see what I say to you, it was quite slow at the beginning of the 20 years ago, but now we are in a big acceleration because there is some international programs that is dedicated to sequencing specific type of plants. And it was 5 years ago now, programs have announced 1,000 medicinal plants need to be sequenced for their genomes. So international community share which plant need to be sequenced. Okay, some are very common, but some are more specific of specific species, specific families. And that could be interesting because it misses a lot information of lots of small plant family, plant medicinal family, for example. So perhaps in 10 years, we will have more thousand new genomes. And this is, yes, I like this one from 2200 and from now 2024. Here are not the 1,400 plants that have been sequenced, but the more important one that have each year here, some of them, for example, here, the mice, have completely changed the view we have of how functions the maize genome, for example. So it was important because all of these plants have been entirely sequenced and lead to several very important discovery of their physiology, but also their metabolisms, primary and specialized one. Okay. Sorry. One thing to have also in mind is that genomes, obviously, is nuclear genomes, okay? But in plants, you know, you have two other type of genomes dedicated to organelles, plastid genomes from chloroplasts mainly, and also mitochondrial genomes. In genomics, okay, development, we can include also the sequencing of these two types of genomes. They are very interesting because they are smaller than the nuclear one, but they are multi-copy in one cell. So it means you have not only one mitochondria or one plastid, you have more. And in each plastid, you have not exactly the same genomes. You have the genes. All of the genes are present and they are encoding for the same type of protein, but the allele of each genes are different. Means you may have some isoenzymes that could be also interesting to have to know. So when we develop some genomics strategy, we can include also organelles sequencing in order to have an idea of what are the diversity of cytoplasm genomes in plastid or in mitochondria. Another thing also to have in mind, which is quite interesting to know, is that the number of chromosomes in each plant are very different. Here you have some example, and from the number of chromosomes, you don't have plants that have 2n equal 2. We haven't found for the moment plants that have this type of number of chromosomes. The smaller one is 2n equal 4, and until you see here some of plants or 2n equal 600. So you can imagine in this case the amount of DNA that you have in one, okay, nuclear genomes means that you have lots of copy of genes because this number is increasing because you have lots of duplication of the entire genomes. So this is why in some plants, sequencing the genomes starts to be hard, depending on the size of this genome. So you need to have this estimation before you decide to make a sequencing program, okay, for specific plants. And some of interesting plants that live in a specific condition means that they have specific adaptation, so means that may have specific specialized metabolite. Sometimes they have very bigger genomes than more common plants. Okay. And also, the complexity of the genome is due to the repartition of the different type of sequence you can have in the genomes. You have, obviously, genetic part, means this is the sequence that encode, okay, for specific protein. But it is not all the genomes behind this type of sequence. You have what we call intergenic sequence, non-coding sequence, okay? Not intron, it's not intron. It is between two genes, okay? So you have lots of sequence of that. They have a crucial role for regulation of the transcription of the coding sequence, but they take a lot of part of the genomes. And also you have what we call transposable elements. In the genome of plants, there is a lot of sequence that can be copied and transferred in another part of the genomes that lead to several natural mutation and also modification of the sequence of the genomes. And what you can see here is that depending of the plant, here are Arabidopsis thaliana, the smaller one, in which more than 40% of the genome is dedicated to genes that are encoded. And if you look at another Brassicaceae, Brassica rapa, for example, with the bigger genomes, you may have lots of DNA, but only 20 or 30% of the genome is dedicated to sequence, real sequence encoding for specific proteins. So it means that when you make some genomic analysis or genomics, here are the important sequence you have to highlight, but also both of these two parts need to be clearly identified, because in this part, what you can find, it is also lots of regulatory elements that can also have very important impact on the transcription level of all of your genetics, for example. So it means that when you make some genomics, you have not only to identify the genetics parts, but only all the other parts of the genome. And now we have found that some of this not coding part is very, very, very important. And just here I have take a part of one of the picture I have already shown you, just to remember you that the size of the genome can be very different from this, the smaller one. And you see here, for example, Nicotiana bentaniana, which is a plant very useful for biotechnological development, have one of the biggest genomes in size, and also a very high amount of genes that is encoded in these genomes. So this is what we just for you to remember that there is a very big diversity of that. So what happens when we can have access to the entire genomes after is how to, we have identified, I don't know, it was, for example, in Arabidopsis about 25,000 genes, it's the average, okay? And we can try to organize the genes according to the family of the activity of the corresponding proteins. For example, you have some that are involved in cell growth, in cell rescue, in cell defense, but in intracellular transport static energy, protein synthesis, and so on and so on. And some are dedicated to metabolism. And in 200, when the first genomes was published, just a small part of the gene have been dedicated to metabolism. primary and secondary one. Okay. So what we observe now, because it was 24 years ago, okay, we have continued to analyze these genomes, and in particular, what we have observed now is the development of specific method, including what we call machine learning. So the idea of machine learning is quite simple. You make a model, okay? In this model, you enter parameter of which you know exactly the results. And in fact, the machine learns by itself how they can link its artificial intelligence, yeah, okay? So the machine is able to learn how to manage the data you have given to have exactly the answer you are waiting for. And after only, you implement new data, and the machine normally is able to predict different, for example, function of gene. And by this way, after you can control if it is okay, you said yes or you said no, and step by step, the system is able to increase the ability to predict, in fact, okay, for example here, the function of a gene according the sequence and according what have been published for the different sequence and already completely proved, experimentally proved, okay, in different other studies. So here, using always Arabidopsis thaliana models and applying this new methodology, depending of artificial intelligence, we have seen that some papers have shown different models predicting functional activity of each genes. And by this way, it was allowed a lot of new genes for specialized metabolisms, but also for primary metabolisms. That was interesting because, in fact, we thought to know lots of things of primary metabolisms, but instead there is a lot of genes, new genes, that have been also discovered, but characterized to be potentially involved, okay, in this type of metabolisms. And what also it was interesting is that at this moment, metabolisms cover all types of metabolisms. Now, by applying new models, new methods, we can distinguish genes that could be more dedicated to terpenoids pathway, alkaloid pathway, or phenolics pathway. So that could be more precise than was before. But it is true that for these 24 years, we have accumulated also lots of data, new data, okay, concerning this particular metabolic pathway. So you see here that 86% of metabolic, specialized metabolic gene that were not used to create this particular model, when they have been tested with the model, they have been predicted in a good way. So this is the way to test the robustness of the model, which is always important in this type of strategy. And in this condition, they have found 1,220 new genes that were not described as potential gene involved in specialized metabolite pathway. But now we don't know which are the enzyme and which are the pathway in which they are completely involved and so on. But we have a base of new genes and each of them can be studied in order to see in which metabolic pathway it could be involved. Okay. So in 2024, I saw a very nice paper in BMC Genomics. You can go to take a look. It's quite complex. I don't want to explain all of the step because it is a model developed with a multi-omic feature, what was not the case until now, means that in fact always from Arabidopsis thaliana, instead of trying to find gene only for one type of metabolite families, they share from 3 features, terpenoids, alkaloids, and phenolics. So they develop different models, and in order of this model, we are able to confirm, okay, the role of well-known genes involving both of these 3 features, but also they propose really new genes, okay, that could be distributed also in these 3 type of feature. And what it is also very interesting to see in this paper, very strong one, is that they have also tested different other plants with this model, new models of analysis of genomes, and they have shown here the robustness also of these new models. They have also tried tomato, and I don't remember another type of organism, but it is very that have a very high number of sequence. But they have also applied this to less known plants, and they have shown that this model could be really developed to predict new genes. Okay? Don't ask me the detail of the model. I will be completely unable to explain this. I appreciate only the results because it could be for us a very successful tool. And by this way, I have in my own team some bioinformaticians that are very, very expert in this type of development, and they could take care about this new model to see how to apply, for example, for our own research. And when I am in contact with other international teams that have exactly the same type of works, not on the same pathway, but on other pathway, this type of paper are very important for developing, okay, new strategy at a lab level, okay, in order to develop knowledge of a metabolic pathway. So for you, it's not necessary also to understand all the detail, be sure of that, just to know that it exists. And each year you have some very strong paper showing new modelization way, new algorithm. Okay, so means that now this science is really close to more informatician, modelization science, computational science that have been shown during the conference, but at one level that is becoming very, 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 very strong. Okay, so now it's quite difficult to be an expert in all the field needed to control in order to find this new metabolic pathway. And what it is interesting by this way is also that some new pathway, completely new pathway can be discovered even in a plant in which you know for a very long time several pathways. Because some of metabolites are difficult to analyze. So you can miss a part of metabolite in metabolomic study. As I said to you, it's a snapshot, so you can completely miss a large part of metabolite. But with genomics analysis, as it is a complete library of information, you can, by this model, discover new potential metabolic pathway that could explain also why, at what moment you observe this metabolite and you can't link this metabolite to all the other you already known, okay, in the cell. So this is why I wanted just to share with you this type of strategy, because we can have some new hypothesis of metabolic pathway, of how some metabolites are produced. And by this way, I think in the future they will be able to discover a new metabolic pathway. So just to inform you what are the techniques that are now well developed. So there is some new generation of sequencing that are used for genomics or for transcriptomics. Firstly, it was Sanger's method. I suppose that all of you have already heard about this method. It's quite an ancient one that it is not used now. And now what it is more used are this one, what it is called second generation sequencing, NGS, that it is very fast because in fact you take all the genomes, you fragment it by ultrasound method. It's a mechanical one and with depending of the frequency and the time you applied ultrasound, you can fragment the DNA in smaller or in bigger fragments. That is very interesting because you can apply different strategy after. And these fragments are directly amplified and after sequenced. Or now there is a new generation of sequencing named third generation sequencing that don't need to amplify all the fragments. Fragments are directly sequenced. So it is faster than it was also by these techniques. What means faster? It means that in fact, here it's a sort of shaders, okay? When you have cut the DNA, make sequence, you can, in several hours, you can have an idea of how genome could be assembled. And after it need only 1 or 2 days to overlap all the sequence you have obtained to make contigs, means long reads, okay? And after that, when you have all your contigs, you may need, they say 30 minutes, you see it's quite fast, okay, to test the quality of your assembly of the genomes. And after several days can be also dedicated to make some structural analysis of the genomes you have sequenced. So means that in 1 week, at maximum perhaps 10 days, that's all, you may have all the information of a new sequencer's genome. But it is really, really fast, okay? And the cost is very, very, very low, okay? You can apply for less than 10,000 euros. So I don't know how many it is in rupees. You have to multiply it by 80, I think, something like that. Okay, so it is considered as not so expensive, okay, analysis of genomes. Okay, so after you can make some draft of genome and you can do what you want with this genomes. And here it is another website because one of the conference have show you some website in which you can find several information. This is one of the most complete website of all the plants that have been sequenced. You can see here that in this color, bright green, it is the plants where genome have been only sequenced one time, but you see here somehow have been sequenced twice or more than twice. And this is the very useful website for us to have access to all of this data because it's a free access, okay, website. It's very important to have this type of website, and it was shown you also because, in fact, the community, the scientific community can manage better this data when they share the data with the others instead of taking all the data for themselves. Depends. Some companies, for example, that have a very strong R&D department are able to sequence a lot of plants, and for the moment they maintain them secrets. But at what moment they will be needed to share with scientific community because it's very difficult to manage all this data alone. Okay, you are looking for something specifically, but the genome can be used for developing different other type of science. So it's interesting to see that an international community can share all of this data for developing a very nice science, not only for metabolic pathway, but for different plant science too. So here, I have time? Okay.
Yeah.
Yeah.
We have 5-10 minutes before we break, and then we can continue. I think it's 5 minutes to 11.
Yeah.
So do you want to continue? I was thinking maybe we can break in 10 minutes from now, and then we can continue in another half an hour.
I can just introduce, and after we can stop and we can continue. What I want to illustrate here is what we can do at a lab scale. It is, for example, the workflow we have completely developed in my own laboratory, but with also with my other collaborators, in order to be efficient to discover new genes according the use of omic data. So it's more concrete, perhaps. So here the first step is obviously to identify the plant and the metabolisms. You want to have more information. For us, for example, it's alkaloids, monoindole terpene alkaloids of Catharanthus roseus, that it is one of the main pathway we are studying for. And for example, the idea is now this pathway it starts to be really well known, but it misses already some specific enzymes. So what it is interesting is that you can reconstitute a metabolic pathway, as I show you, by the knowledge of 1, 2, 3, or you see 4 enzymes, and it misses, for example, this one. So one of the first steps is to identify what type of enzyme could catalyze specifically this type of step. It is already the strategy I showed you before. We do the same. And for example, here we suppose that the missing activity that can permit to transform D in E, okay, will be supported by, for example, a P450, sorry. Okay, so by this way, what we can do after? We can, this is the hypothesis. So the second step of this workflow will be to try to find what are this type of enzymes, cytochrome P450, okay, that is produced in our plants. We have two types of strategy, or we can directly predict the gene from genomics data. But in this case, we have all the P450 gene encoding this type of protein. But you have seen that, for example, in some genomes, you may have hundreds, okay, copy of genes that encode this type of enzyme. So it could be difficult to manage. So what it is useful to use is to have transcriptomic analysis, because we can find conditions in which this specific molecule F is produced. So it means that in this situation, all the enzymes necessary to do the pathway will be present, and also the transcript. So it is simpler, in fact, to conduct this type of analysis on RNA sequence instead of genome sequence. So by this way, we can cover only a part of the genome. So we have less gene candidates, okay, at the end. When I say less, it can be several, 10, 20, 30 candidates, which is quite too much, okay? We can't verify each candidate if we have too many candidates. So what we can do after to prioritize, in fact, in this candidate, which could be the most probable candidate than the others. This is perhaps the more difficult choice of the strategy we have to develop in order to decrease the number of candidates. For example, there is four different strategies. One, it is named homology-based screening. I will show you exactly what is it. The second one is random mutagenesis. The third one is the physical gene clusters. It is what I have spoken before, or co-expression analysis. We have this both strategy possible to develop. What means by them? The first one, homology-based screening, use exactly what I have shown you before. We use the literature, all the sequence of the gene that have been already known in literature in other type of plants that screen homology to orthologous genes, and by this way we can distinguish several small sequences that are well conserved, and after against our transcriptome or against our genomes we can blast this sequence to the transcriptome or genome of Catharanthus roseus in order to isolate specific candidate gene. But in fact, what we have already remarked for several times is that we can have lots of negative candidates, because in fact you can select enzymes that catalyze not this activity but other type of activity. So sometimes it's not so, it's easy, it's quite fast, but not so secure to explore the candidate. So after we can use another type of strategy named random mutagenesis. In this case, we can mutagenize plants by UV, by different chemical approach. And after we screen all the plants obtained after mutagenesis in order to select those that are not able to produce the metabolite we want. It's sort of a silencing, of the production. So by this way, we can after make some new RNA sequence in order to know where is the mutation, and after we can try to have an idea of if the mutation is in a specific gene encoding this type of enzymes, it could be probably the candidate. It's a very powerful strategy, but very laborious too, because to obtain mutagenized plants, it's quite long. To make some new sequence, transcriptome sequence, in order to compare the sequence to find where is the mutation could be very long, and it costs a lot too. But if no other strategy is functioning, it is a possibility to have access to the information. And the third one is the physical gene cluster in the genome. So the idea is, that's all the enzymes that are involved in a pathway are present physically, on the same part of the genome, on the same chromosome. This is very common in prokaryote genomes, more rare in eukaryote genomes. But in fact, it was now from several years that some of clusters have been identified for specifically secondary metabolisms. The first one was in maize, it was from specific metabolite in maize, and from this date there was a lots of study aiming to find if in each genomes we can find this type of cluster of genes that are dedicated for a metabolic pathway. But what it is true is that in fact it was not so often until now. We don't know all, and only about 20 gene clusters involved in a different biosynthesis pathway, have been already identified in plant kingdom. But what it is interesting is that as we have seen that in the nature, clustering the genes in a small portion will lead to a functional metabolic pathway. This is a way to make some artificial new metabolic pathway for engineered system that will be able to produce exactly the metabolite we wanted. So it opened the door for synthetic biology strategy, and I will show you that in the future. Here, it's for you. You could see it. It was the first and the other. It is the list, sorry, of all the cluster genome that have been already described and proved. You can see here that it is in different type of organs for different type of compounds, triterpenes, diterpenes, but also cyanogen glucoside, for example. In different plants also, in Arabidopsis thaliana for sure, but also you see different other type of plants, including evidently sorghum and maize. Papaver somniferum, which is also a medicinal plant for some specific alkaloids, okay? Unfortunately, we haven't found some in Catharanthus roseus until now. We are always looking for, but it seems that all the metabolic families may have some clusters in several plants, but as you know, we haven't sequenced all the medicinal plants, so we are sure that in the future we will be able to find new cluster of this type, which is really interesting. So here, for example, it's a nice paper that has been published this year, the last cluster identified in maize, okay? It concerns always the biosynthesis pathway of benzoxazinoid, okay? And what it is interesting is that, in fact, a large part of the gene involved to the production of these particular intermediates have been already observed in this chromosome, chromosome 4, okay? And you can see that quite all the metabolic pathway all the genes, encoding enzyme of the metabolic pathway are situated in the same part of the chromosome. And it is funny because, in fact, it's a long time we are working on maize. And in fact, this cluster have been already partially characterized 20 years ago, but now we have been completely formalized. And also they have made lots of nice works making the gene with also regulator genes, such as encoding, for example, for transcriptome factor, transcription factor, that could be also linked with this cluster. So you see that there is a very nice evolution of this domain of the study in order to have more tools for the future to identify new pathways. So these methods can be very interesting because if you suspect the existence of the cluster, you can, and if you have the genome sequence, obviously, you can take a look near the genes you have already identified to see if there is some that could encode enzyme of the family you are looking for example. So it can be quickly a way to reduce the number of candidates, but you need to find a cluster. And the last one, it is what we have already discussed a little bit together, the co-expression analysis. This is metabolite. This is based on the hypothesis that all the genes need to be coordinately expressed in order to have a functional metabolic pathway, okay? And by this way, you can have some of the non-genes, for example, the 3 first gene as bait. You can study what in the candidate are the genes that are co-expressed. So it is transcriptomic analysis, okay, to know if in this gene some are closely expressed or not with all of the gene of the metabolic pathway. So only the enzyme, the genes coding the enzyme you are looking for that are co-expressed with both of the genes of the metabolic pathway will be really more strong candidates for the enzymes you are looking for, okay? So you have different strategy, okay, to make this type of study of co-expression. You make some statistical comparisons, okay, and with it is what we call identify differentially expressed genes. You can construct some co-expression networks. It's another strategy, computational strategy, to see how all of these genes are co-expressed, in which you can also add some metabolomic data. The main statistical system used are the Pearson correlation coefficient. I don't know if here there is some statistician. Perhaps? Or no? I am not also a statistician, but I use a lot this type of tools. Okay, so it's quite easy to build a little bit long, but it's quite easy. And you also, if you have the chance to have some bioinformatician with you as collaborator, you can also unsupervised machine learning techniques, develop this type of technique. Why it is called unsupervised? Because we don't know the knowledge of gene function. Okay, we enter with no a priori. Okay, in this type of strategy. So you can see here we may have lots of way in order to reduce considerably the number of gene candidates to 1, 2, 3, or 5 at maximum candidates. At this point, it's quite easy to make some what we call validation of function, or by producing the recombinant enzymes and test the recombinant enzyme by in vitro assay, okay? Or we can also develop some functional strategy in vivo, which is more interesting also, by making some knockout plants for these specific genes. And that's a way to prove that if you disrupt the activity of the gene, you have no metabolite produced. So it is a correlation between function also and presence or no presence of the protein. So I will show you the different techniques we can use for this. Okay, and then after that, you have discovered new genes and you can make a very nice publication. We make a break, and after I show you 2 examples of this strategy applied for cataract cirrhosis, for example. Okay? Thanks for your attention.